All right, so welcome to another episode of Inside IALR. Thanks for tuning in today. So we have a lot of, I'd say, interesting research, complicated research, and definitely research that is meant to be applied very soon. And that's our whole division of research here is called applied research. Today we're here to talk about biotechnology, which is a very, very cool concept, very cool term, but also a very important industry, both for what's happening right now, and then as far as in the future as well. So I'm here with Dr. Scott Lowman, our Vice President of Applied Research. I'm here with Dr. Sajeewa Amaradasa, who's our Senior Scientist here, and Dr. Chuansheng, who is our Chief Scientist here at IALR. So thank you all for joining us today. I really appreciate it. So to start, I'm just going to open it up to all three of you. Tell me a little bit about what is biotechnology? What is that term? What does that term mean? And then tell us a little bit too about sort of what is your research focus in that? What is your specialty in biotechnology?
Well, first of all, at the most simple level, biotechnology is just using a living organism like a yeast or a fungus or a bacteria to make a product or a process. We've been using these for thousands of years. Think about wine, cheese, and beer. But more recently, in the last century, we used it first, I think, to make antibiotics for World War II. My area of research is really focused on more molecular biology and microbiology. I don't do a lot of research now. I spend most of my time developing the program, but I was trained in molecular and microbiology, and I earned my degrees from William & Mary and Virginia Tech.
My background is plant physiology and molecular biology. Yeah, after I work here, I focus on bacterial endophyte, only for beneficial bacterial endophyte for plant growth promotion. We try to develop the low import and sustainable agricultural system.
I'm a plant pathologist by training. So here at IALR, I study bacterial endophytes for antifungal properties. I try to use them in biocontrol of diseases in economically important crops.
Thank you all for that introduction and sort of that, big picture, what you all focus on. And you all touched on it a little bit, but I kind of want to get a little bit more into it. Is so our where the work that you all are doing is part of our Plant Endophyte Research Center, which endophytes, I'll let you guys explain those because you can explain them way better than I can, but just very generally plant bacteria that we're taking those bacteria, have a library of them and are using them to help plants either respond to stress or to grow better. Tell me a little bit more about the Plant Endophyte Research Center, and specifically, you are talking about, Dr. May, you're talking about, those bacteria that help plants grow, whereas Dr. Amaradasa, I know you're more looking at how do you help them respond to those stress or fight off pathogens. So tell us a little bit about that side of things.
The Plant Endophyte Research Center is focused on developing biologicals to reduce or eliminate the use of pesticides and fertilizers. Fertilizers damage the environment. Think about streams and other things like nitrification and global warming. And pesticides have harmful human health effects, as well as many as have been banned over the years. Our research center is set up in labs that are full-service biology labs, so we're able to take the bacteria from the inside of plants and characterize them based on how they can help the plant in the case of biostimulants, or control diseases in the case of biocontrol.
Think of synthetic pesticides. Yes, they are effective, but there are a lot of disadvantages, like environmental pollution. They kill non-target pests, and there is even the target pests can build resistance. So over time, these pesticides become ineffective. So one solution is to use biopesticides. They are naturally occurring, and therefore you don't see any resistant buildup, and they are environmentally friendly. So what I do is I use my library, this bacterial collection, and do a plate assay in the lab to understand whether they can control the plant pathogens, and then I use those good ones in the greenhouse to test them in plants. in a nutshell, that's what I do.
That's a good explanation. I have the smaller version of that in my head, so now I'm going to remember that explanation you just gave.
Another bigger area for beneficial bacteria is plant growth promotion, also called biostimulate. So this bacteria, has some plant growth promotion traits. For example, they produce plant hormone, IAA, and phosphosolubilization, and nitrogen fixing, and also enhance stress tolerance.
Just a little more background on biologicals in general. We know in human health that there are things called probiotics. They've really only come to prominence in the last about 20 years. It's the same thing that's happening in agriculture. For decades, agriculture has been dominated by the chemical industry. So chemistry or chemical companies developed molecules that they could patent, and then they sell those for a period of 10 or 15 years. So they made a lot of money on those. So the industry is really built on chemistry. But as we move forward and talking about sustainability and keeping pollution out of streams and human health concerns, a way to address that is through biologicals. And even some of the largest chemistry companies are now turning to biology and partnering with us in many cases to look at how biology can be used to cure these problems and to address consumer demand for clean food.
That's another very good breakdown of. I hadn't thought about. Because I know we talk about biotechnology and biologicals here, but I didn't realize that was the shift, that we're going from chemistry to biology.
Yeah. In many cases, biology is a lot more clean. To grow a bacteria, it likes kind of like the same temperature humans like, about 70 some degrees. The pH is relatively easy. It's not a harsh environment. When you talk about chemistry, you're talking about harsh environments when you're mixing chemicals together to form reactions. So it's a more sustainable way to even produce a product.
And this research that we're doing, I know, as we said up top, where it's applied research. This is not research that's meant to be potentially useful 10 years down the road. This is all research that we're doing here that's meant to be useful right now. So tell me a little bit about, and I know Dr. Amaradasa, Dr. May, you guys have both published papers within the last year. Peer-reviewed publications with our team here. So we are doing, real scientific research that's going out in peer-reviewed journals and things like that. But what is the application of the research that we're doing now, both, in the immediate and then even in the next few years?
Okay, one of area we want to, focus on phosphate. And actually, we published one paper, 1 year ago. So that paper, we screening some good bacteria to solubilize phosphate. Because in the soil, there's lots of phosphate, but only 1% there's compounds is available to plants. Most is just insoluble. Plant cannot use it. And the farmer usually use more soluble phosphate. And plant only maybe take 20-30%, and other will become unavailable to plants. And some of run off to river and contaminated to water resources. Some algae bloom in a lake. So this we want to find some good one and use this good one so farmer can immediately reduce use phosphate fertilizer. Yeah, that's one of example.
Think of controlled environment agriculture. These days, hydroponic crops are getting, like leafy vegetables are getting more and more popular. So I did a test in hydroponic lettuce. Pythium is one of the most important diseases in hydroponic lettuce. And we don't have any labeled pesticides for hydroponic crops because it's a new thing. It is catching up these days. So I used one of our good biocontrol endophytes to see whether it can control Pythium in hydroponic lettuce. I first added the biocontrol endophyte. It is a 1619, the strain name. And then later on, I introduced the pathogen. So I found that our biocontrol endophyte can increase the fresh weight, I tested 2 cultivars. In one cultivar, it increased the fresh weight by 29%, the other one by 42%. So by that example, you can understand the potential of using biocontrol endophytes, bacterial endophytes, to enhance, to increase yields without using traditional pesticides.
And to add to that, one unique thing about our center is that we can pivot quickly. So we just got into controlled environment ag research only a couple of years ago and have already made discoveries about how to use biologicals in that setting to increase growth and yield. Another great example is our work with strawberries. So we had Cooperative Extension come to us a few years ago. Their growers were losing a important soil fumigant called methyl bromide. Well, it's a really nasty chemical that was banned. But strawberry growers were relying on it. And then once they lose that chemical, their yields go down about 15 to 20%, almost immediately. So this is very important for strawberry growers in Virginia. Cooperative Extension came to us with that problem. We teamed up with Cooperative Extension, screened our library for biocontrol properties for these pathogens that are important to strawberries, right, that target strawberries. Did our work. We got a grant from the Department of Ag to do further work, and we basically found that we were able to increase marketable yield by about 14% with those organisms from our collection. Those organisms are now in the hands of companies trying to commercialize those. In the case that Dr. May mentioned, the phosphate solubilizing bacteria are also with our partners now, because we can only take something so far here at the Institute. We can't really scale up, manufacture, sell, distribute a product. We really have to rely on partners to do that part of it.
Let's keep going on down that train of thought about what our role is. So what you're saying is that we work with a lot of partners, and I think that's true across our entire organization. There's very, very, very little we do that's by ourselves. We're always working with partners, whether that be, from everything from the federal government to private companies to state government, local, all of it. We're working with different partners. But what is, I guess, kind of big picture, how are we working with, and you just outlined it a little bit, Dr. Lowman, but what are some of the different ways that we are working with partners as far as biotechnology? What is our role in that?
So one way we work with companies is contract research. So that's intellectual property clean. We just agree to do an experiment for a company and then give them the results. So they get a high quality third party validation of their product. Another way we work with companies is through partnering with the companies to develop IP together. So a company may come to us and want to use bacterial endophytes for a product. We can help them develop that, and that benefits us in the end if we get a licensing agreement, and then there's money generated from that. Another way, we rent lab space. So we have companies here on site. We have 2 companies currently, more on the way, that are using our facilities to grow their own companies. So they use our skills, their expertise. They rely on and partner with their scientists, but they also have their own scientists here working to create products. So those are primarily the 3 different ways we work with companies: contract research, IP co-development, and providing lab space.
And we're working with dozens of companies at any given time, right?
Yeah, currently we have over 80 NDAs, non-disclosure agreements, signed with different companies doing different projects. Those include people like Canon, who we've had a press release. So Canon, the camera people, are working with their plant scientists to develop models of plant growth and health. We work with biotechnology and chemistry companies to help them enter the biologicals market. And we even provide polymer lab space and testing and analytical chemistry services.
Especially startup companies need our data because we are an independent research institute. They can, in return, go back with our data and ask for more grant money. So in that way, we help many startup companies to develop their biopesticides.
And as far as to go, big picture, and Scott, you mentioned, or Dr. Lowman, you mentioned this in the beginning, the idea that, at cheese and alcohol are examples of biotechnology at the most, fundamental level. But now, what are some of the I guess, the main industries that biotechnology is being used in? And I understand most of ours revolves around agriculture, but what are some other? Where is this industry going? What industries are using biotechnology?
So two things. Currently, the biotechnology market worldwide is about $1 trillion. Most of that work is in the area of human biology and biotechnology, so biopharmaceuticals. So a lot of research and effort is going into that area. But one of the fastest growing areas is ag biotech, so these biologicals we're talking about, but also food and beverage. You've heard, everybody's heard, to some people it's horrific that they're growing meat in labs. Well, that's biotechnology. They're using cells to produce meat. But it's expected that this century, biotechnology is really going to expand and basically explode in production, because we have more tools now than we had even 30 years ago. The types of next generation sequencing, the way we can manipulate organisms, biotechnology, and particularly fermentation, can be used to make plastics. They can be used to make food enhancements. They can be used to make all sorts of things. And really the sky's the limit as far as what they can produce. So in the future, there's tremendous growth ahead. So it's a great time to be in this sort of at the leading edge at this time.
And I want to go back to the Plant Endophyte Research Center. So I know, we have a library that's, like the first thing said on our website. We have a library of about, 2,000 bacteria. Where did these bacteria come from? How did, I know they're naturally occurring, but I mean, where are we getting these bacteria? What is that characterization process like? How are you deciding which ones might be good for which research?
This library, probably, we started maybe 5 years ago, Scott and I, to want to expand our program. So we isolated this bacteria from natural plants that grow in nature and healthy. And after isolate, we store in -80, make stock. And then
-80 is cold.
Yeah,
just
cold.
For our listeners at home.
Yeah. So after that, we need to characterize each one. We see is called plant growth promoting traits, including plant growth hormone, auxin, IAA, phosphorusylation, and nitrogen fixing, ACC deaminase, siderophone. So after we calculate all this, and then we do some plant growth promotion test in vitro, so eliminate the other factor. So after that, we move to greenhouse, and right now we work on in a CEA center. So this long process,
And we chose native plants because they haven't been selected by humans. A lot of people may not realize, but like a plant or a crop like corn has been selected for thousands of years. It's been manipulated basically by humans, picking the largest and the best and those type of things. So we wanted to get a snapshot of Mother Nature. What does Mother Nature put inside of plants that live in the wild, that don't need fertilizers, they don't need pesticides, they're perfectly fine on their own. So that's where we started, and then where we went from there was developing assays, and that really makes our collection different than most. Most companies and people simply genetic sequence their bacteria or their fungus. Well, there can be lots of genes present. If they're not really doing something, it's not valuable to the bacteria. So a bacteria could say it has the ability to produce auxin, but if it's not being turned on genetically, that bacteria will probably produce no auxin. What we do that's different is that we have assays that measure how much auxin is being produced. So we measure what's happening that the bacteria is actually doing.
Actually, we also use in vitro growth promotion test. We use Tofesco grass in a magenta box. This is called in vitro test. So somehow we can get 500 time increase, because this is in vitro condition. That's a common problem is, after move to field, the promotion disappeared or dramatically reduced because the environment is totally different.
Yeah, I guess that's a principle in life, is it's easier to produce results in a lab or a completely controlled environment than when there's lots of variables involved.
So that's why recently, maybe three year we got 2 published because we're working in a CEA center. This is CEA controlled environment is somehow like in vitro test, in vitro condition. So we find some potential application in a CEA. Usually in a field trial, somehow you get the inconsistent results. Yeah, definitely.
Because there's so many other variables at play that...
Because natural condition, change too much.
Right. Dr. May, you mentioned, we've been working on this for about 5 years. We have a library of these 2,000 endophytes. What's sort of the growth, what are we expecting to do next? Or in 5 years from now, do we want to be able to say we have a larger library of endophytes? Do we want to be able to say many of these are licensed by companies and being used by companies? I mean, what's sort of the goal of the Plant Endophyte Research Center?
The goal is definitely to get these out to market. Mm-hmm. So to work with partners, to help startups grow, but to license these out to generate income for our research program, but also help improve the environment, help meet the needs of consumers, and more.
I think it's really cool. Very simply talking about it, we're taking naturally occurring bacteria that are, found in nature, as you guys have described, taking those, figuring out what they do, how they might help a plant, putting them in another plant and seeing positive results. And that's really cool that those are naturally occurring, and not having all those negative environmental impacts that are out there. And even Dr. May, what you're talking about with the phosphate, that you're helping a plant access what's already there. It's basically trying to help that plant access phosphate that's already available to it, just making it so the plant can actually obtain it. That's just really cool stuff. But I think that's all the questions that I have. I think, this research is, it sounds super fancy, sounds like it should be not for right now. And in some ways it doesn't sound, I'm not a scientist, so to me all of this sounds like science fiction, but it sounds kind of futuristic in some ways, but I think it's not. What you guys are talking about is companies are already, working with us on using these in their products right now. But is there anything else that you all would want to add, or anything else you think it's important that people know?
Just say that you sort of nailed it there, that relying on biology is something that humanity did for thousands of years, up until really the 1960s and the Green Revolution. So after that, there's a lot of chemistry involved. So we're really just returning to our roots and leveraging what Mother Nature supplies. Just one more thing. Our next steps is we're getting more into fermentation. So we've purchased a fermentation device, a bioreactor they call them, and we'll be doing a workshop before too long in partnership with Virginia Tech to train people on how to use that fermentation device. But we're also at the same time we're members of the Virginia Biotechnology Association called Virginia Bio. Now we're members of Charlottesville BioHub. The Roanoke Blacksburg Technology Council also has a sort of a bio focus. And we're really trying to help create an ecosystem. Those other three that I mentioned are really focused on human biotechnology. Well, a big opportunity is in ag biotech and food biotech. So we hope to position ourselves to be able to attract more companies like we're already doing to come to this facility and grow through biotechnology.
I want to add something. So we are only testing, as a plant pathologist, I'm testing only endophytes, bacterial endophytes. That is just one tool in the biocontrol toolbox. But there can be other microorganisms that we can use to control diseases like fungi or beneficial other nematodes like that. So there are a lot of areas that we have not investigated. So the potential is very large.
I just want to emphasize maybe there are really good potential application in a CEA.
Yeah, no, I think the research happening in the CEA Innovation Center it's cool to watch, cool to see it. Obviously, I'm not there every day, but just cool to see it's a cool place to look at for one thing, and it's definitely some interesting stuff going on there. So, thank you all for joining us. We really appreciate it.
Thank you. Thank you.
Thank you.
Megabytes, IALR's on-site cafe features everyday favorites, convenient ordering options, and an eager staff ready to welcome and serve you. Our enticing menus offer something for everyone, from a fresh, well-stocked salad bar, wraps, sandwiches, and our rotating hot buffet offered on Tuesday, Wednesday, and Thursday. Megabytes is open Monday through Friday from 9:15 a.m. until 1:30 p.m. To view menus and for more information, visit ilr.org/megabytes.